Vanakkam friends, I am Dr. Vanangamudi. I come back after 3 months. Really, I was suffering from a lot of pain in the right hand shoulder, not only in the bones but also the muscles because of continuous usage of um, computer, especially laptop, for many years. Now also, I am feeling very little pain. However, I am much interested to present video and interact with you from now onwards. And let us coming to the topic of this presentation. The topic is seed dormancy and what are all the types of dormancy that is available in different kinds of seed, and as well as in the beginning I would like to present uh, about the different terminology that are in seed dormancy. As I told earlier, I was working as a professor and head Department of Seed Science and Technology, Tamil Nadu Agricultural University, followed by the Dean, Adivarasakti Agricultural College, Kalavi, that is a private college affiliated to Tamil Nadu Agricultural University. And before retirement, I worked as the Dean Agriculture of Agriculture College and Research Institute, Tamil Nadu Agricultural University, Coimbatore. I retired from service in August 2013. Now we can discuss about the different terms as well as the definitions. What do you mean by dormancy? It is the general meaning is the lack of growth due to external or internal issues or causes or the factors. Really talking about the seed dormancy definition, this is failure of viable seed to germinate even if you give favorable environmental conditions like water, oxygen, temperature, and so on and so forth, and the suitable medium. The next term is quiescence, and this is the condition in which seed can germinate upon absorption of water, but there is no internal germination barriers exist. And next term is the after ripening. It is a physiological process that occurs in the seed after harvest that enables germination to take place after the harvest, that is the post-harvest period. And there are two types of dormancy. First one is primary dormancy, and this type of dormancy develops when the seed is attached to the mother plant itself, and it exists when the first harvest is over. And the second type of dormancy is the secondary dormancy, a condition that develops to prevent germination after the seed has been detached from the mother plant and then exposed to specific unfavorable environmental conditions like temperature and the light. And the endogenous dormancy, and any cause of dormancy that is related to the embryo, that is the internal part of the seed. The best example is immature development of embryo or endosperm or chemical inhibitors that are available inside the seed. And the next one is exogenous dormancy. This is a mechanical resistant type. Maybe the physical impermeability to the water, and there may be the availability of some inhibitors and light sensitivity associated with the seed coat. This is mainly related to the seed coat. What type of dormancy? Exogenous dormancy. Whereas endogenous dormancy is the internal part of the seed is playing a major role in the dormancy induction. And the double dormancy or combined dormancy, two or more dormancy types occur in the seed. Example, the fleshy or dry fruits with chemical inhibitors that combines with hard endocarp. The typical example is teak, where the endocarp is very hard and the pericarp, that is a fibrous nature, that is having some kind of chemical inhibitors. And the thermodormancy, this is the secondary dormancy. These types are the primary dormancies, exogenous, endogenous, and double dormancy or combined dormancy. And thermodormancy is the secondary dormancy. This is a type of dormancy induced by high temperature during germination of seed, while the photodormancy, it is mostly influenced by the light, and such seeds are therefore also called light sensitive seeds. And this type of factor, when it is imposing on the germinating seeds, this kind of a photodormancy is being developed. And there are two things, the phytochrome, red, and the phytochrome for red. And the red light, the 660 and 760 nm, don't forget the several questions, the paper, the competitive exam, they can ask this type of questions. Whereas the white light is otherwise called as far red, and the red, the ratio is 2:1. And the far red light having the intensity of 760 to 800 nm. If the seed is having the phytochrome red, that is dormant in nature, when it is converted to far red and the dormancy induction is being released, and that becomes germinable, that is called as non-dormant seed. Now we can discuss one by one the different types of dormancy that are existing in the plant kingdom. And the first one is primary dormancy, and in that the first kind is exogenous dormancy. We have discussed in the earlier slide, and this is the factors that are related to the outside the embryo, mainly the seed coat. And seed coat, some of the species is facing the physical barrier for the entry of water as well as oxygen. And another kind of seeds, they will exist, they will prevent the the emergence of embryo, and that is called as mechanical dormancy. And this type of dormancy is also not allowing the water get inside as well as oxygen. The chemicals, mostly the inhibitors. For the best example is coumarin in the coriander seeds, that is just the spice seed. And the second type of dormancy in the primary dormancy is endogenous dormancy, and this dormancy is mainly related to the embryo itself. And again there are three types, that is morphological dormancy, and this morphological dormancy is having three types: rudimentary, linear, undifferentiated, maybe the underdeveloped or immature embryo. And the second is physiological dormancy. This physiological dormancy may be again subdivided into non-deep, intermediate, and deep. And the the another type of dormancy is combinational, that is double dormancy. There are two types of dormancy exist in some of the seeds. I can repeat once again. The primary dormancy, the dormancy that develops in the seed when it is on the mother plant itself, that is before harvest. And this exogenous having three types: physical, mechanical, and chemical. Endogenous dormancy, it is related to the embryo. This is also primary dormancy. And another subdivision is morphological, rudimentary, linear, and differentiated. Physiological, non-deep, intermediate, and deep. Third type of dormancy is combined dormancy. These are all primary dormancy. Now we can discuss about the secondary dormancy. There are two types: thermodormancy and photodormancy. And the exogenous dormancy may be the physical condition as discussed earlier. The seed coats are impermeable to water and oxygen. The best examples are most of the legumes. The cause for the physical dormancy is the structure of the outer cell layer, which becomes impermeable to water, and the macroscleroid cells, a mucilaginous outer cell layer, or a hardened endocarp are the main reasons that seed coats become impermeable to water, not only to water but also the entry of oxygen, which is more important for the germination of seeds. The second type of primary dormancy is the mechanical dormancy, and the seed coats are too hard that will allow the embryo to expand during germination. The causes: structure of seed coats or remaining fruit occurs. This classical example is olive tree. In nature, the seed coats are softened by environmental agents such as acid in guts. When the Acacia nilotica curvale seed, the pods are being taken by the goats, and in the system, the stomach, the acid is hydrochloric acid is developed, is secreted, that aids in the digestion of the food material that are taken inside the goats. And likewise we have similar mechanisms, and because of acid scarification, when the seeds are coming in the droppings of goat, that will ready to germinate. And the second type of the mechanism that can eliminate this kind of dormancy is microorganisms, the warm or moist environmental condition, and forest fire. This is a very suitable treatment for the teak in the forest area. If the forest fire occurs in the teak plantation, and the next season, when the rain is receiving, the teak seeds is ready to germinate. And the different treatments that are available to overcome the mechanical dormancy is the abrasive action. We have to abrade the seed coat of the seeds either with the sandpaper or with the sand, and likewise the scarify with sandpaper, and hot water treatment. And the temperature of the hot water may be around 90 degrees Celsius. And when you do this treatment, and you should not put the seeds when the water is on the fire. We have to take out the water from the fire source, and then the seeds are poured. And the next thing is acid scarification. And most of the legume seeds are given with this type of treatment. For 1 kg of seed, 100 ml of commercial grade sulfuric acid is needed, and you should not use the dilute sulfuric acid. And this sulfuric acid, when it is diluted, the acid is also get absorbed along with water by the seeds, and that will damage the internal part of the seed. And the another type is fire and use of immature fruits. Before reaching harvestable maturity, you have to harvest, and that immature fruits can be germinated to overcome this type of dormancy. And most of the treatments, whatever we discussed here, the main cause, the main mechanism is softening of seed coat, hard seed coat. That is more important. So that makes the seed to absorb water along with oxygen easily. Once scarified, water can get in. And one thing have to be careful because the seed is also more susceptible to damage by microorganisms. And the chemical dormancy, the presence of chemical inhibitors in the outer coverings of many fruits and seeds that will induce dormancy. It occurs, this type of chemical dormancy occurs mostly in fleshy fruits, hulls, capsules, and many dry fruits. The classical examples are apples, citrus, grapes, tea, coriander, spinach. As I told earlier, this is having coumarin, the toxic compound, the inhibitor. And how to overcome this chemical dormancy? Simply you soak the seeds in the water and then leach out. In the running water or either in the irrigation channel or in the tap, have a bundle of seeds, and that will be kept in the water, running water. So easily most of the inhibitors are washed by the running water. And even you can expose this type of chemical dormant seeds in the heavy rain, and to counteract the toxic effect of the chemical inhibitors as well as nullify that, you have to treat with the hormones, especially gibberellic acid 3. And either you can go for chilling for a few days. Then endogenous dormancy. First one is morphological dormancy. Embryo is not fully developed at time of ripening. So we need additional embryo growth after the seed is separated from the plant. And the classical examples are ranunculus, poppy, woody species such as holy date palms, often combined with other kinds of dormancy such as hard seed coats. So the combined dormancy or double dormancy types exist. And we have 3 types in the morphological dormancy. Rudimentary means pro-embryo stage. Before the embryo is reaching the full maturity, and it stops its development and maturation in the pro-embryo stage. And the examples, the members of the Ranunculaceae family, Papaveraceae family, and Araliaceae family. And second is linear type, and again there is a stoppage of the growing embryo at torpedo stage. And the members of Umbelliferae, there is a spelling mistake, Ericaceae and Primulaceae. Undifferentiated type of organs that occurs in the morphological dormancy. The classical example is orchids. And to overcome after ripening, you have to allow the seeds to dry or some period before that is supplied for sale or for sowing. That period is after ripening. As soon as it undergo after ripening period, most of the underdeveloped embryo and endosperm will develop and reach maturity. And alternate warm and cool temperatures. And the third type of dormancy breaking treatment is the application of soaking of seeds in the gibberellic acid. The extended period at high temperatures, the date palms often takes several years to reach full embryo development and germinates, even 2 years. And often high temperatures, 100 degree Fahrenheit, will accelerate this type of dormancy. And the second endogenous dormancy, second type is physiological. That first one is non-deep. Non-deep means often short, termed 1 month to 6 months, the type of maturity. Example is lettuce that disappears when the seed is stored. And there are two kinds, the secondary dormancy. So far we have seen the primary dormancy, mechanism involved, the causes involved, and how to break the dormancy of primary type of dormancy in different kinds of seed. And the second type of dormancy is the secondary dormancy. There are two types, photodormancy and thermodormancy, may be due to inhibitors as well as control of gas exchange, that's especially the oxygen. But this light requirement can be substituted by cool temperature growing the seeds in the dark below 70 degree Fahrenheit. Non-deep, to overcome these hormones, you have to soak the seeds in hormones, and again you have to counteract with abscisic acid and kinetin GA3. And photodormancy appears to be through phytochrome. As I told earlier, sunlight is having the red and far red ratio of 2:1 that results in phytochrome in the active form. If the ratio is 2:1, that is an active form. Under foliage, that is under shade, the red and far red ratio is 0.122:1 or 0.7 to 1. That is, far red dominates, resulting in phytochrome in the inactive form. Okay. And the intermediate, the control mechanisms to be in seed coat and tissue around embryo. Example, most of the temperate seeds of temperate trees and conifers. Embryo itself is quiescent, non-dormant, and germinate if excised. If you remove the seed coat, the seed will germinate. Excised embryos exhibit physiological dwarfing and stunted growth, rosette-like structure. To overcome this, you have to give chilling treatment, that is a cold stratification for 1 month to several months, exposing pre-moistened seed at 0 to 5 degrees Celsius in the refrigerator, and it can also be substituted by GA3 treatment. Deep dormancy, to overcome this, you can allow the seeds for natural stratification and maybe the moist chilling, pre-soaks in water 50% moist and kept at 35 to 40 degree Fahrenheit. And there is one thing you have to note it, the temperature is too high, secondary dormancy is induced, then another chilling period is required. So kindly avoid high temperature while you do the moist chilling. For apple, the temperature is 70 degrees Celsius, 17 degrees Celsius, or the 62 degree Fahrenheit. Secondary dormancy prevents the seed from germinating after it absorbs the water. Main causes are temperature extremes, prolonged darkness, prolonged light, water stress, and oxygen extremes. I have tabulated all the types of dormancy. The first column is dormancy types, characters, example, and dormancy breaking stimulus treatments, and one is natural and another is seed handling. If it is immature embryo, that is physiologically immature embryo, Example fraxinus, ginkgo, and the post-dispersal development, that is after ripening. And if you give artificially, then it is after ripening. Mechanical dormancy, the embryo emergency is restricted due to hard seed coat, pterocarpus, and most of the terminalias and melias, and decomposition of hard structure by termites, mechanical cracking of the hard seed coat, that is abrasive action with against the sandpaper or the uh, concrete mixer also. The physical dormancy, imbibition is impeded due to the impermeable seed coat, most of the Leguminaceae family and Myrtaceae family. Abrasion by sand, high temperatures, and exposing the seeds to high temperature, either water soaking, hot water soaking, ingestion by animals, that is acid type of method. And the mechanical scarification, boiling water or acid scarification. And the chemical dormancy, chemical inhibitors, compounds that prevent germination, and most of the fleshy fruits like berries, drupes, and pomes, leaching by rain or decomposing by fruit pulp, removal of fruit pulp plus leaching with water. Photodormancy, this is a secondary dormancy. Seeds fail to germinate unless exposed to appropriate light regimes. The temperate species like Betula, humid tropical pioneers, Pathodia, and Eucalyptus, exposure to white light or red light likely to promote germination. Exposure to light during germination, light dark cycle of variable duration. Thermodormancy, temperature controlled dormancy. Examples are Fagus, Quercus, Pinus, tropical and subtropical pioneers. Exposed to low winter temperature, bush or forest fires, diurnal fluctuating of temperature. This helps naturally to break the dormancy. If you go for artificial, that is stratification or chilling, kiln or light burning, fluctuating temperature. With this, I am completing this presentation, that is seed dormancy. I elaborately dealt all the types of dormancy, including different terminology and the mechanism, the breaking treatments, different treatments to break the seed dormancy. I hope that you will enjoy with this presentation and you can learn more with this. Thank you very much for my listening, and kindly subscribe my YouTube channel to get the upcoming videos, and kindly share this presentation to your friends as well as your group. Comment on my presentation. Thank you very much, friends.